Good afternoon, everyone. Thank you for your time and interest, and thank you for the organizers to give us the opportunity to present Vivet and some development. We've been incorporated in July 16, and actually we recently been funded with a series of 37 million, and very nicely we've been awarded by Fierce 15 as one of the interesting companies, and I think it's a great challenge for the team and for the patients we're actually targeting some very severe diseases, basically. An introduction to Vivet in a nutshell. We basically ask us at the origination of the company, so what's going on with the gene therapy and what are the main issues? We all know that part of the issues is the manufacturing, and there's some of the things we can and we cannot do. We're not a manufacturing facility, but we have to make sure we get the right strategy in terms of manufacturing. But in the meantime, we all know there's the sustainability of expression we need to handle, especially if we target the children, especially in some of the diseases we're targeting. And also we need to understand what are the immune responses and the way we could actually improve the immune tolerance to the AAV to make sure we can treat children and eventually think about how we could actually re-inject the patient. Therefore, to do that in liver, even if we have some kind of tolerogenic liver, we need to have a very specific liver and liver AAV with a specific tropism. And therefore, what we did is we had a very specific strategy for having a new AAV, engineered AAV called NKT. So the structure of Vivet is really around this specific partnership we have with CIMA, which is a center developed in Pamplona, and where we actually do all of the research, and I will introduce you to the team. We also have this specific license around this Massachusetts RNA AAV NKT from Luc Vanderberger lab. We're doing all of this new technology platform for sustainability and immune response. We're subcontracting clearly the manufacturing, but we're ensuring that all of the manufacturing will be able to start from the process development to the commercial facility to avoid any transfer, because we know transfer, even if we have some potency assay, is not without any significant risk. We're subcontracting all the non-clinical development. But more interestingly for us, we clearly try to understand and really have an in-depth research around the famous 3P, which are the physicians, the payers, and the patients. Basically, that works. So the team is based on an experienced team I'm coming from GeneSight, and Sanofi work. But more importantly, I have the team for the Chief Scientific Officer, Gloria, in Spain. She has more than 20 years experience. And we have people coming from Novartis and Anokyo, as Jens Kurt is the co-founder. And we have people from experienced Chief Medical Officer for the team of Genzyme and General Counsel and Finance as well. So that's important. We get a team who cover all the different strategic assets of the company, from merger and acquisition, for example, to the science, basically. So the investors who actually supported the Series A financing are Novartis Venture Fund, Roche Venture, ElseCap, and we have two Spanish funds where we have an affiliate and as Columbus Partner and Izios and Kurma Partners. So what is CIMA? Maybe you don't know CIMA. CIMA is a center of investigational medicine who has a real translational approach with the faculty to actually combine the synergy between the clinical facility and the research program, but also benefiting from this hepatology approach to a transversal gene therapy combination with all of the platform. And that's very important for us because we have a team as part of the research collaboration of more than 15 people working over the 4 different indications. So why did we target some liver disease overview, basically? These inherited liver disorders are actually targeting, and when you look at the complexity of this disease, what is very important to understand that 50% of the people affected are children. And when we look at what are the main curing approach on some of the disease is liver transplantation. When you think that in the US, approximately 12.5% of the children less than 18 years old are actually part of the representing the liver transplantation. But more interestingly, if you look at the distribution of this series of liver transplantation in adults and in children, you can see that Wilson disease is representing approximately 10% in the children, less than 18 years old, but almost 19% in the adult patient. But also some other diseases like progressive familial intrahepatic cholestasis. And that's very important because that's also a necessity that you can cure, and that's approaching the rationale for the disease. So what is Wilson disease? Wilson disease is a simple, in quotes, autosomal recessive inheritance disorder with a prevalence of 1 to 30,000 births, and a prevalence, for example, in France of 1.5 to 100,000. And it's a mutation, it's various genotyping mutation in an ATP7B transporter. We are all eliminating the copper through the bile, and if we can't, there's a very significant accumulation into the liver and clearly also into the brain with some significant impact. So basically, if you're not able to transport the copper with the support of ceruloplasmin into the bile, you get accumulation, and no treatment definitely lead to the death of the patient. So Wilson disease is characterized with a wide clinical range that can actually be present with hepatic symptoms, significant elevated liver enzymes, chronic hepatitis, and some patients are diagnosed so lately that they declare an acute liver failure and go directly into transplantations. The other very significant impact is the neurological outcome and psychiatric outcomes. They can be presented signs of Parkinsonism, coriform, tremors, dysarthria, and actually they have some complexity in the speech approach. So the diagnosis is made of a combination of different elements, but there's a clear need for improvement because some of the patients are really diagnosed really too late. It does exist some treatment, but in the meantime, these treatments have a very significant issue is compliance. Not only they have some severe side effects, like 20 to 30%, but on top of that, the copper actually still increase and evolve. There's still have an evolution of a disease, and eventually some treatment initiation lead to neurological deterioration in 20 to 50% of the patients, which are irreversible. So clearly there's a high need of a new treatment, knowing that more than 10 to 15% will go into liver transplantations. So when we discuss with KOL, and that's important to understand what would be the position and interest of such gene therapy, for them it's clearly a very likelihood of success in the notion that it replaces the liver transplantation. That's the main asset for them, independently of the genotype. So it's phenotypical cure for them. When you think about the kind of patients, you can actually treat the adult patient, stabilize, but also the patient without cirrhosis, which can be stabilized patient on treatment, which represent in some ways the majority of the patients, despite they still have some lack of compliance and evolution of complications. Initially, we'll think about the pediatric patients and also the one with mild neurological impaired patients. Why? Because some of the patients who had transplantation has neurological recovery. Even if it takes some time, so over 1 to 3 years, they have some significant benefits. And lastly, it would be the exclusion for the moment as a patient with severe inflammation of the liver, because doing gene therapy in such patient would be probably useless, while if you can actually also treat the pediatric patients. But as some of the patients we need to control and treat over time. In the meantime, we have to think about the payer side, and the payer side clearly indicates that we need to demonstrate in kind of superiority to the standard of care. At least equivalence to liver transplantation would be a great asset. We need long-term, and that's the part of the sustainability of expression, and also the patient care total cost saving. That's why we all need to do this kind of burden of illness, cost of patients, and if you include the liver transplantation, that's a significant approach. And the clinical suitability, as they call it, is very important. That's why you need to rationalize the type of patient you're going to treat. So based on this and the knowledge we have over the gene therapy program that has been published in Journal of Hepatology and deserve an editorial, that's interesting to see that the knockout mice model clearly shows some very significant improvement, not only for the biological marker, but also from the structural and histological point of view. Firstly, we use a new AAV. This AAV-NKT is an engineered AAV, which actually is being made by the Luc Vanderberg team. And you can see that there's a more homogeneous distribution in the liver, but more importantly, in the non-human primate, there's a superior transvection. Even if it's a small number of animals, we're currently exploring this later, and that's clearly confirming the interest that it's superior transvection with less pre-existing immunity compared to AAV8. So based on this data, we actually explore VTX801 in the with a truncating ATP7B gene, for the main reason that the wild-type ATP7B is pretty large, and you may use the total capacity of the AAV cargo if you use the hot total gene size. So what we did is an optimization. It's like a mini gene approach, and we clearly demonstrated, because the wild-type gene didn't work in female mice, that actually there's superior efficacy in female compared to the wild-type. And we clearly demonstrated in a dose-response manner that we reduce the copper hepatic content. We increase the serum ceruloplasmin, which actually lead to the activity in the serum and transport of the copper to the bile. And there's also, importantly, two different things to look at, is the evolution depending on the stage of disease, and also the excellent safety profile. So if we look at the approach of treating animal at different stage of the disease, it's interesting to see that we got a similar efficacy between 6 and 12 weeks old animals, and using this in male and female patients, in female mice, sorry. And the disease phenotype is really corrected. So it's really an approach in terms of homeostasis of the copper. And if you look at the liver enzyme on the top left, you could see that we really restore the normal values with a wild-type truncated T2 form, which is a product we're developing, and also reduce the copper elimination in urine, because that's the alternative way if we don't have any bile elimination. And also, that's a very important marker, is the improvement of ceruloplasmin transportation. So clearly, this genetic transfer of ATP7B clearly reduces the liver damage, control the urine excretion, and improve the ceruloplasmin, restoring this full homeostasis of copper, leading to a transportation-like approach. So based on future discussion, we currently have discussion with Europe and US, we intend to start by Q4 2018 a phase 1 program that will lead to a pretty classical dose escalating, but also treating adult female and pediatric patients. But more importantly, that we're going to be able to look at the copper metabolism over the 6 to first 12 weeks using specific technology at PET scan imaging, for example, and showing us the real modification of the homeostasis of the copper metabolism, supporting the transvection level in the liver. And that will help us to clearly define the dose. We've received the orphan designation in both in Europe and US for this product. In addition to this major indication for us, we're developing this NKD platform and the other sustainability of expression in 3 other indications, and that's PFIC2, PFIC3. So PFIC2 and 3 are what we call progressive familiar intrahepatic cholestasis. It's a deficiency in transportation of the bile acids, would actually lead to some hepatocarcinoma and lead to very severe liver disorders, leading that to the fact that 100% of the PFIC2 patients will have liver transplantation in less than 20 years old, and 60% of the PFIC3. And clearly, that's something very important. So we're currently developing the animal models, and we'll get the results by mid-2018. And similarly for citrullinemia, we're working on a transgene vector construct optimization to treat not only adults, but to treat children. And that's very important because in this patient population, managing hyperammonemia, for example, lead to very low IQ patients, less than 90 or 100. And for example, the cost of a crisis of ammonemia is about $500,000 when you get hospitalizations. So being able to get this in a sustainable expression manner is very important. So we're supporting this with the CMC platform of the NKT and baculovirus production scales, and the IP, and also the common platform on using NKT with the regulatory support. So in a nutshell, the global portfolio is leading to a Wilson indication with an IND and initiation in Q4 18, proof of concept in mid-2018 for the other 3 indications, and amplifying the platform of immune response modulation and transgene expression sustainability to apply this new development over these 3 other indications. So interestingly for us, when we look at what is necessary to actually support this development of gene therapy in children, we thought it was very important to leverage our platform and design for complementary gene therapy in indication with high unmet need, currently not addressed. And you need to have an enhanced sustainability, even if you only show results over 1 or 2 years. but we know we have up to 7 years expression today. And also manage the immune tolerance approach. That's something very important because we'll be able to actually re-administer potentially, even if we have a window within the first 2 weeks, but actually control the immune tolerance for all of the patients. So thank you for your attention.